So all of you, we start with plant hormones. Every year NEET exam में they ask you some parts of plant hormones. So 5 plant hormone है: auxins, gibberellins, cytokinin, ethylene, and abscisic acid. They will ask you in the NEET exam something about these plant hormones. So let's analyze these plant hormones in detail. Let's begin with this. Yeah, so let's derive these plant hormones. Just a second. You have auxins, you have ethylene, you have cytokinin, you have abscisic acid, and you have gibberellins. ये 5 hormones है. सबसे पहले, where are they all derived from? Auxins are derived from the amino acid called as tryptophan. Auxins are made from tryptophan. Auxin याद रखना किसमें से बनता है? Tryptophan. Ethylene is also derived from an amino acid called as methionine. तो ये सब आपके एनसीईआरटी में लिखते भी जाना, ये बहुत इंपॉर्टेंट है, बहुत बार पूछते हैं नीट एग्जाम में. ऑक्सिन्स किसमें से बनता है? अमाइनो एसिड ट्रिप्टोफैन. ethylene is derived from methionine. Cytokinins are derivatives of the purine base that is adenine. तो कौन सा हार्मोन डीएनए की मदद से बनता है? If they ask you from NEET point of view, which hormone is associated with nucleic acid? तो डीएनए में क्या होता है? एडेनाइन. और एडेनाइन की मदद से बनता है साइटोकाइनिन. And then you have abscisic acid, which is a carotenoid derivative. Carotenoid, we remember, are plant pigments, photopigments. So you have chlorophyll A, chlorophyll B, carotene, xanthophyll, photosynthetic pigments है. उन photosynthetic pigment carotenoid से बनता है abscisic acid. So if they ask you in the NEET exam, which hormone is a derivative of pigments of plants? The answer is abscisic acid. It is a derivative of carotenoid. And gibberellins are chemically terpenes. Gibberellins are chemically terpenes. तो ये 5 चीजें याद रखना. अमाइनो एसिड डेरिवेटिव कौन से हॉर्मोन्स है? अमाइनो एसिड डेरिवेटिव ऑक्सिन्स एंड इथिलीन. ऑक्सिन कौन से अमाइनो एसिड से बनता है? ट्रिप्टोफैन. इथिलीन कौन से अमाइनो एसिड से बनता है? मिथियोनिन. Which is the DNA based or nucleic acid based amino acid? Cytokinin. It is made from the DNA purine base called adenine, which is a plant pigment based hormone. Abscisic acid. It is a carotenoid derivative made from carotene and xanthophylls found in the क्लोरोप्लास्ट इन द क्वांटोसोम्स. उस एब्सिसिक एसिड का डेरिवेशन कैरोटेनॉइड होता है. एंड जिब्बेरेलिन किस में से बनते हैं? जिब्बेरेलिन्स आर मेड फ्रॉम टर्पीन्स. जिब्बेरेलिन्स आर मेड फ्रॉम टर्पीन्स. ये याद रखना.
बंद करो दरवाजा.
Please put yourself on mute, all of you. And cytokinins are derivative of adenine. The hydrogen atom at the N6 of adenine is substituted by benzyl, that is benzyl adenine or furfuryl to form synthetic kinetine or isophenyl group to form natural zeatin. तो साइटोकाइनिन 2 टाइप के होते हैं. आई होप यू हैव रेड इट. सिंथेटिक काइनेटीन. made from herring, fish, animal cytokinin is zeatin. Animal cytokinin is zeatin. और प्लांट में नेचुरली क्या प्रेजेंट होता है? सॉरी, एनिमल साइटोकाइनिन इज काइनेटीन. एनिमल साइटोकाइनिन इज काइनेटीन. और प्लांट्स में नेचुरली क्या बनता है? जियाटिन. तो हाइड्रोजन एटम ऑफ द 6th नाइट्रोजन बेस ऑफ एडेनाइन अगर बेंजाइल और फॉस्फोराइल ग्रुप से सब्स्टिट्यूट होता है, देन इट फॉर्म्स काइनेटीन, which is the animal cytokinin. अगर वो आइसोफिनाइल ग्रुप से सब्स्टिट्यूट होता है, then it forms zeatin. This is given in the NCERT. याद रखना, N6, 6 नाइट्रोजन ऑफ एडेनाइन अगर बेंजाइल और फ्यूरफ्यूराइल ग्रुप से सब्स्टिट्यूट होता है तो बनता है काइनेटीन, और अगर आइसोफिनाइल ग्रुप से सब्स्टिट्यूट होता है then it forms zeatin. ये याद रखना. And very importantly, auxins, यहां बाय मिस्टेक ये भी है, auxins, gibberellins and cytokinins are growth promoters. Abscisic acid is the growth inhibitor, and ethylene is both, but it is mainly an inhibitor. So, which hormones are growth promoters? They help in growth of plant. Growth promoters are auxins, gibberellins, and cytokinins. They all help in growth. So, auxin, gibberellin, and cytokinin are growth promoters, which is growth inhibitor, which will inhibit growth. So, it is antagonistic. It is abscisic acid. Abscisic acid is growth inhibitor, and which has got dual functions. Means it is both growth promoter as well as growth inhibitor. Then the answer is abscisic acid. Dual functions, both promoter and inhibitor. Then that is ethylene. Ethylene. I'm so sorry. तो ग्रोथ प्रमोटर कौन सा हो गया? Auxins, gibberellins, and cytokinins. These are strictly growth promoters. Growth inhibitor कौन हो गया? It is abscisic acid. And dual function, both growth promoter and growth inhibitor, कौन सा हार्मोन हो गया? Ethylene. ये भी याद रखना. And abscisic acid 3 टाइप का होता है. इनहिबिटर बी, एब्सिशन 2 एंड डॉर्मी. ये याद रखना, इनहिबिटर 2, एब्सिशन 2 एंड डॉर्मी. ये याद रखना. एंड नाइस पॉइंट्स गिवन इन द एनसीईआरटी. Tips of coleoptile in canary grass grow towards light because auxin was stated by Darwin. Tips of coleoptile in canary grass grow towards light because of auxin. In other words, coleoptile tip light की तरफ जाता है, which is called as phototropism. Phototropism. And phototropism यानी growth towards light कौन कराता है? Auxins help in growth towards light. Auxins help in growth towards light. तो ये याद रखना, auxins help in growth towards light, which is called as phototropism. And Cousins showed that a gas causes banana ripening, and Gain proved that ethylene is synthesized by plants and ripened fruits. Which is the gaseous hormone? Gaseous hormone was proved by Cousins that ethylene is a gaseous hormone. And where is ethylene produced? By plants and ripening fruits. So ethylene helps in the ripening of fruits. Ethylene helps in the ripening of fruits. ये याद रखना. Which hormone helps in ripening of fruits? Ethylene helps in ripening of fruits. And ethylene causes a downward rolling of leaves, which is called as epinasty. ये आपके एनसीईआरटी में दिया नहीं है, इसे याद रखना. Which hormone causes downward rolling of leaves, which is called epinasty? Ethylene causes downward rolling of leaves, which is called as epinasty. तो सर समझो लीफ बहुत ज्यादा सनलाइट को एक्सपोज्ड है, तो अगर लीफ ऐसे सनलाइट को एक्सपोज्ड है एंड सनलाइट कुछ ज्यादा ही आ जाएगी, देन लीफ अगर ऐसे रहेगा, देयर इज अ हाई रेट ऑफ ट्रांसपिरेशन, ट्रांसपिरेशन के चांसेस बढ़ जाएंगे. So ethylene will cause downward rolling of the leaf, so exposure to sunlight will become less and chance of transpiration will decrease, and that is called as downward rolling of the leaves, which is called as epinasty. Which hormone causes epinasty? It is ethylene. So which hormone causes phototropism? Phototropism is caused by auxins. And which hormone causes epinasty? Ethylene causes epinasty. ये कुछ नए points किए. Now we go into all the hormones. Auxin, the meaning of auxin, it is ready to grow. And auxin was first isolated from human urine. तो सबसे पहले ये सब NCERT points है. Which is the hormone isolated from human urine? From human urine, auxins were first isolated from a patient having a disease called as pellagra. तो pellagra disease जिस patient को होती है, उसके urine में से auxin पहली बार isolate किया गया. Auxins are the most common type of naturally occurring plant growth hormone. सबसे common plant growth hormone कौन है? Auxins. Auxins are the most common plant hormones, and they were discovered from oat coleoptile by a scientist called Wendt. Wendt discovered it from oat coleoptile. And auxins are synthesized in roots, shoot tips, young leaf primordia, and they help in elongation. Auxins are synthesized in roots, shoot tips, young leaf primordia, growing leaf, and they help in elongation. And auxins are weak organic acids produced naturally by plants and are capable of cell elongation. Auxins are used up during the growth process, so they are not catalyst. Other hormones only act as a catalyst. Auxin actually ग्रोथ, सेल डिविजन, सेल एलोंगेशन, ग्रोथ के अंदर यूज हो जाता है. सो इट इज नॉट अ कैटलिस्ट. याद रखना, ऑक्सिन्स आर इनएक्टिवेटेड बाय लाइट. लाइट के प्रेजेंस में ऑक्सिन इनएक्टिवेट हो जाता है. विच हार्मोन इज इनएक्टिवेटेड बाय लाइट? ऑक्सिन्स. And auxins are translocated in a polar manner from apex to the base and in the roots upward stem downward. तो auxins ऊपर या नीचे polar जाते हैं. If they go from base to apex, it is called as, if you've heard of it, acropetal. And from apex to base, if it goes down, it is called as basipetal. एंड ये सिर्फ ऊपर और नीचे जाते हैं, इनका साइडवे मूवमेंट होता नहीं है. तो साइडवे मूवमेंट होता नहीं है, दैट इज कॉल्ड एज पोलर मूवमेंट्स. ओनली ऑक्सिन्स शो पोलर मूवमेंट्स. ऑल अदर हार्मोन्स शो साइडवे मूवमेंट्स. ओनली ऑक्सिन्स शो पोलर मूवमेंट. दे गो अप एंड डाउन, एंड दैट इज कॉल्ड पोलर मूवमेंट ऑफ ऑक्सिन्स. तो पोलर मूवमेंट इज सीन एक्सक्लूसिवली इन ऑक्सिन्स. Auxins are natural or synthetic. Naturally occurring auxins are indole 3 acetic acid IAA, indole 3 butyric acid IBA. So IAA and IBA are naturally occurring auxins. Synthetic auxins are NAA, 2,4-D and 2,4,5-D. NAA, that is naphthalene acetic acid. Naphthalene acetic acid. And you have 2,4-D, that is 2,4-dichlorophenoxyacetic acid. And 2,4,5-T, that is 2,4 2,4,5 trichlorophenoxyacetic acid. तो NaA 2,4-D and 2,4,5-T, naphthalene acetic acid, 2,4-dichlorophenoxyacetic acid, and 2,4,5-trichlorophenoxyacetic acid are synthetic oxidants. So IAA, indole acetic acid, IBA, indole butyric acid are natural oxidants. Synthetic oxidants are NaA, 2,4-D and 2,4,5-T. ये याद रखना. Lower concentration of oxygen promotes the growth of stem, but higher concentration of oxygens are required for growth of stems. Sorry, lower concentration of oxygens help in growth of roots, and higher concentration of oxygens are required for growth of stem. तो रूट ग्रोथ के लिए थोड़ा सा ऑक्सीजन चाहिए, स्टेम ग्रोथ के लिए ज्यादा concentration ऑक्सीजन का चाहिए. And Wendt performed something called as the Avena curvature test in coleoptile tips of oat. And Avena curvature test is called as the bioassay test of oxygen. Bioassay test of oxygens was done by Wendt, and Wendt did something called as Avena curvature test in coleoptile tips of oat, Avena sativa. तो दो साइंटिस्ट ने वर्क किया है. Wendt worked on coleoptile tips of oat, Avena sativa, to give the Avena curvature test, and Darwin worked on canary grass and showed that canary grass grows towards light, which is called as phototropism of oxygens. Phototropism of oxygen. तो ये दोनों याद रखने हैं. Wendt did the Avena curvature test in coleoptile tip of oat, Avena sativa, and Darwin did canary grass growth towards light, which is called phototropism of oxygens. ये दोनों याद रखने हैं. What is the role of oxygens? Oxygens help in cell division, also help in cell elongation. In tissue culture, oxygens help in the formation of callus. तो ये आपने टिश्यू कल्चरिंग में किया है, ध्यान से सुनना. आई एम डिस्क्राइबिंग इट अगेन. समझो आपको एक परफेक्ट प्लांट मिला है, और परफेक्ट प्लांट में एक्सेलेंट फ्लावर्स, वेरी गुड स्टेम, लॉट ऑफ वुड अवेलेबल इन द स्टेम. तो आप एक परफेक्ट प्लांट का क्या करोगे? यू विल डू टिश्यू कल्चर. टिश्यू कल्चर में क्या करोगे? कट द रूट टिप एंड कट द शूट टिप. रूट टिप and shoot tip are full of meristematic cells. Root tip and shoot tip are meristematic cell. उसको एक टेस्ट ट्यूब में डाल देते हैं जिसके अंदर होता है न्यूट्रिएंट मीडियम. से कोकोनट मिल्क इज पुट. कोकोनट मिल्क डाल देते हैं और उसके अंदर रूट टिप या शूट टिप डाल देते हैं. एंड कोकोनट मिल्क विल प्रोवाइड शुगर, nutrition, electrolytes, vitamins, everything required for cell division. फिर उसके अंदर डालते हैं oxygens and cytokinins. Oxygens and cytokinins are put in an equal dose. Equal dose में oxygen cytokinin डालते हैं. It will make fantastic cell division, and that root tip या shoot tip will grow into a big mass. And that big mass is called as a callus. Big mass is called as a callus. फिर callus के ऊपर समझो ये callus है. Callus was formed by equal oxygen and cytokinin. Oxygens equal to cytokinin. Then we got callus. Now we put extra oxygen. More oxygens are put inside. And as you put more oxygens, callus will start developing roots. Callus में से roots develop होते हैं with the help of extra oxygen. Then we put extra cytokinins. तो extra cytokinins डालेंगे, callus में से shoot develop होगा. साइटोकाइनिन्स एक्स्ट्रा डालेंगे, इट विल हेल्प इन द फॉर्मेशन ऑफ शूट। तो ऑक्सिन कैलस में ऑक्सिन डाला, व्हाट विल इट हेल्प इन फॉर्मेशन ऑफ रूट्स। और कैलस के ऊपर साइटोकाइनिन डाला, व्हाट विल इट हेल्प इन फॉर्मेशन ऑफ शूट। इन अदर वर्ड्स, स्टेम इज पुट इनसाइड। इस तरह से टिश्यू कल्चर किया जाता है। फिर याद रखना, गिवन इन द एनसीईआरटी, ऑक्सिन्स हेल्प इन जाइलम डिफरेंशिएशन। तो ऑक्सिन मेरिस्टेमेटिक सेल पे एक्ट करके it will help in xylem differentiation. Auxins like indole acetic acid and naphthalene acetic acid, when applied to cuttings in lower concentration, help in adventitious root formation. तो जब वेजिटेटिव प्रोपगेशन करते हैं तो छोटा सा रूट टिप या शूट टिप लेके उसके ऊपर ऑक्सिन डालेंगे, it will help in adventitious root formation. Therefore, auxins are used in artificial vegetative propagation by stem or leaf cutting. समझो एक स्टेम का कटिंग आपने, बनाना को कैसे ग्रो करेंगे आप? बनाना में सीड तो नहीं होता है, तो बनाना का एक स्टेम कटिंग लेके उसको जमीन में डालते हैं, उसके ऊपर ऑक्सिन्स डालेंगे तो उसमें से रूट्स बनेंगे। या ब्रायोफाइलम में लीफ हैज इन इट बड्स व्हिच आर कॉल्ड एपिफाइलस बड्स। तो ब्रायोफाइलम के लीफ को एपिफाइलस बड की मदद से कट करके जमीन में डालते हैं, उस पे ऑक्सिन डालते हैं, और ऑक्सिन्स विल हेल्प इन रूट इनिशिएशन। ऑक्सिन्स हेल्प इन रूट इनिशिएशन। तो which hormone helps in adventitious root formation? Auxins help in adventitious root formation. ये याद रखना। The presence of apical bud inhibits the growth of lateral buds, which is called apical dominance. And lateral buds require lower concentration of auxin for their growth. Supraoptimal high concentration of auxin suppress growth of lateral buds. High concentration of auxin suppress the growth of lateral buds, which is called as apical dominance. तो ये भी एक अच्छा कंसेप्ट है सर। ये है स्टेम एपेक्स, एंड स्टेम एपेक्स में बनता है लॉट ऑफ ऑक्सिन्स, और ये है अपाइकल बड, और ये है एडवेंटिशियस बड्स। तो अपाइकल बड हेल्प्स इन शूट फॉर्मेशन, and adventitious buds help in stem formation. Adventitious buds help in stem formation. तो इससे stems बनते हैं, और stems बनाने के लिए you require the hormone cytokinin. तो शूट बनाने के लिए कौन सा हार्मोन चाहिए? शूट बनाने के लिए you require auxins, or branches, which is the lateral branches formation require presence of cytokinin. तो याद रखना, स्टेम एपेक्स में से शूट कौन बनेगा? Shoots is made by auxin, and that is from apical bud, and adventitious bud से branches बनते हैं. Who forms branches? Branches are made by cytokinin. But जब तक apical bud produces a lot of auxin, जब तक apical bud बहुत ज्यादा auxin बनाएगा, auxin will go in a polar manner. याद है auxins go in polar manner. वो basipetally नीचे जाते हैं, and they will go into the adventitious buds or axillary buds, and not allow axillary buds to form branches. So auxins will not allow axillary buds to form branches, and that is called as apical dominance. इस phenomenon को क्या कहते हैं? Apical dominance. तो याद रखना, apical dominance is done by auxins. Apical dominance is done by auxins. And then we have auxins prevent the action of hydrolytic enzymes in abscission layer, which is formed at the base of the petiole and prevents abscission. तो ये आपके एनसीईआरटी में दिया है, ये है लीफ, और ये है लीफ. लीफ के अंदर होता है something called as abscission zone, and auxins act on the abscission zone and prevent abscission zone from breaking. Who will break the abscission zone? Abscisic acid breaks the abscission zone, and ये zone टूट जाएगा तो लीफ गिर जाएगा. Who will prevent, who will oppose abscisic acid? Auxins, and prevents falling of young leaves. Young leaves नीचे गिरने से बच जाते हैं. तो auxins, याद रखना, given in the NCERT, promote falling of old leaves. Promote falling of old leaves, but prevent falling of young leaves. Prevent falling of young leaves. In other words, oppose abscisic acid. To oppose abscisic acid and prevent falling of young leaves, fruits, etc. So who promotes falling of old leaves? Auxin. But who prevents falling of young leaves? Auxins, by opposing abscisic acid. ये याद रखना. And auxins are used to produce seedless or parthenocarpic fruits in plants like grapes, oranges, apples and tomatoes. अगर grape, orange, apple, tomato इनके ऊपर auxin डाल दिया without fertilization, the ovary will form fruit. Ovary forms fruit. So ovary will form fruit, but ovules have not formed seed. Ovule का seed बना ही नहीं है, because ovule में seed कब बनता है? Post fertilization. Post fertilization, ovule के अंदर male gamete, female gamete will form zygote. Zygote will form embryo, and the embryo is formed inside, and ovule gets converted into a seed. So what will happen if fertilization takes place? On fertilization, ovule will form seed, but auxins क्या करेंगे? हमें एप्पल में सीड नहीं चाहिए, खाते टाइम वो बीच में आता है, या ग्रेप्स, ऑरेंजेस, ये सब के अंदर सीड नहीं होगा तो वो फ्रूट ज्यादा अच्छा होता है क्योंकि सीड मुंह में आ जाता है. तो व्हाट डू वी पुट ओवर ग्रेप्स, ऑरेंजेस, एप्पल्स एंड टमेटोज? ऑक्सिन्स. Auxins will help in formation of ovary into fruit without fertilization. Without fertilization. So ovules don't form seed. Ovules don't form seed. And तो ovary which forms fruit, fruit के अंदर सिर्फ ovules रह जाएंगे, seed नहीं बनेगा. And such fruits are called as seedless fruits, also called as parthenocarpic fruits. इन fruits को क्या कहते हैं? Seedless fruits, also called as parthenocarpic fruits. And 2,4-D and 2,4,5-T are potent weedicides. They selectively kill dicots and spare monocots. They are used to kill dicot weeds and maintain lawns. तो सब समझो आप एक गार्डन ग्रो कर रहे हो और गार्डन में आप ग्रास ग्रो कर रहे हो. ग्रास इज अ मोनोकॉट, बट उसमें वीड्स ग्रो हो जाते हैं, and weeds are dicots. Weeds are dicots. So what do? We do removal of weeds, mechanical removal of weeds. उससे अच्छा put over this entire garden a lot of auxins. Auxins will, और कौन सा auxin डालेंगे? 2,4-D and 2,4,5-T. Auxins will selectively kill weeds, dicots. Dicots को मारेगा, but it will spare the monocots. तो monocots को मारता नहीं है, सिर्फ dicot weed को मार देता है, and that is the way your entire garden will have only grass. सिर्फ ग्रास ग्रो होगा because 2,4-D and 2,4,5-T have acted like weedicides. And which weedicides, which weeds they have killed? They have killed dicots without killing the monocots. तो ये सारे points ऑक्सिन के फेवर में होते हैं. Then we have gibberellins. Gibberellins were discovered in Japan by Kurosawa in rice plants infected by a fungus, Gibberella fusiculae. Neat point of view, which hormone owes itself to a fungus growth? तो फंगस ग्रोथ कौन से प्लांट से हार्मोन से हुआ है? फंगस ग्रोथ जिब्बेरेला फ्यूजिकोराई से जिब्बेरेलिन डिस्कवर हुआ. तो जिब्बेरेलिन was discovered from a fungus called Gibberella fusiculae. It caused bakane or foolish seedling disease in plants which became very tall and very thin. तो ये Gibberella fusiculae राइस प्लांट में था. राइस प्लांट इज नॉर्मली 5 to 6 feet tall, पर उसके ऊपर जिब्बेरेला फ्यूजिकोराई फंगस आ गया. It became 10 feet tall. And what is a 10 feet tall, very tall plant called as bakane or foolish seedling disease? ये भी नीट पे पूछते हैं, which hormone is associated with bakane disease or foolish seedling disease? Answer is gibberellin. So which hormone is derived from fungus? Gibberellin. Which hormone is bakane or foolish seedling disease? Gibberellin. ये याद रखना. And gibberellins are gibberellic acids, and more than 100 types of gibberellins have been discovered so far, of which the most common gibberellin is G3. G3 is the most common gibberellin, and gibberellins are chemically terpenes. They are terpenes. As we had said before, they are non-polar. Polar up and down कौन सा hormone है? Only auxins. Only auxin. Baki other hormones are non-polar. And what is the role of gibberellins? Gibberellins induce elongation of stem and causes bolting, which is internodal elongation. तो समझो ये है शुगर केन प्लांट, और शुगर केन प्लांट के ऊपर ये है नोड्स, और नोड्स के बीच में ये है इंटरनोड्स. What are nodes made up of? Meristematic cells. Anybody can tell me what are these meristematic cells called?
Intercalary meristem.
Intercalary meristems. Intercalary meristems. And gibberellins act on intercalary meristem and promote rapid cell division. तो इंटरकैलरी मेरिस्टेम का रैपिड सेल डिवीजन होगा, तो व्हाट विल हैपन टू द लेंथ ऑफ द स्टेम? ये है नोड, फिर ये नोड यहां था, अब नोड यहां आ जाएगा, क्योंकि बीच में बहुत ज्यादा सेल डिवीजन हुआ है. तो बहुत ज्यादा सेल डिवीजन हुआ है, लेंथ ऑफ इंटरनोड विल इंक्रीज. योर दिस इज द लेंथ ऑफ द इंटरनोड, और यहां दिस इज द लेंथ ऑफ द इंटरनोड. तो दिस phenomenon of increase of length of internode due to nodes, meristematic cells, which are intercalary meristems, have happened because of stimulation by gibberellin. And इसे internodal elongation called as bolting कहते हैं. Very commonly asked in NEET exam. Which hormone helps in bolting, internodal elongation? Gibberellins help in bolting, that is internodal elongation. Gibberellins help in bolting, that is internodal elongation. And gibberellins therefore cause elongation in jute fibers and also sugarcane. तो सर शुगरकेन प्लांट में मेन काम का क्या है? स्टेम को कट करके उसमें से शुगर केन जूस बनता है. उसका जो लेफ्ट ओवर बगा से रहता है उसमें से पेपर बनाते हैं. तो which hormone increases sugar cane yield? जिबरेलिन इंक्रीजेस शुगरकेन यील्ड। ये सारे एनसीईआरटी के पॉइंट्स हैं। शुगरकेन यील्ड इज इंक्रीज बाय जिबरेलिन। शुगरकेन में व्हाट इज एक्चुअली मेड, okay, what is actually made? Actually gibberellins are made with the help of terpene. They cause internodal elongation and increases your sugarcane yield. तो sugarcane yield बढ़ जाता है. And gibberellins are used to increase leaf expansion. ये आपके एनसीईआरटी में दिया नहीं है, बट समझो कॉलीफ्लावर है, कैबेज है, कॉलीफ्लावर, कैबेज, स्पिनच, ये सब के अंदर हम लोग क्या खाते हैं? लीफ खाते हैं। कैबेज खा रहे हैं वो तो लीफ होता है, स्पिनच खा रहे हैं वो तो लीफ होता है। तो जिबरेलिन स्प्रे करोगे तो लीफ विल एक्सपांड। सो लीफ एक्सपांशन इज डन बाय जिबरेलिन। And gibberellin increases size of parthenocarpic fruits, and they are more effective than auxins to induce parthenocarpic fruits. Which two hormones help in parthenocarpy? Auxins and gibberellins both help in parthenocarpy. Which is more effective than auxins? Gibberellins. So gibberellins are more effective than auxins in parthenocarpy. And Thompson seedless variety of grapes are made by gibberellin. जो हम लोग ग्रेप्स खाते हैं ना, ग्रीन कलर की ग्रेप्स, उसमें सीड नहीं होता है। तो ग्रीन ग्रेप्स that we are eating are seedless grapes. They are called as Thompson seedless grapes, and they are made with the help of gibberellins. Gibberellins help in making of Thompson seedless grapes. And ध्यान से, in case of cereals like barley, wheat, etc., gibberellin treatment induces synthesis of amylase. Amylase converts starch into glucose and maltose available to embryo as food during seed germination. So seed dormancy can be broken by gibberellin treatment. Sir, एक seed के अंदर sugar is stored in the form of starch. We all know that. अगर जिब्बरेलिन ऐड करेंगे एक सीड के अंदर स्टार्च होता है, जिब्बरेलिन ऐड करेंगे, जिब्बरेलिन विल हेल्प इन फॉर्मिंग अल्फा अमाइलेज। और याद है अल्फा अमाइलेज हमारे सलाइवा में भी होता है। व्हाट डज अमाइलेज डू टू स्टार्च? Amylase converts starch into maltose, and maltose becomes glucose. And using that glucose, radicle and plumule will start undergoing cell division. Radicle से root बनेगा, plumule से shoot बनेगा. In other words, seed dormancy टूट जाएगा. So which hormone breaks seed dormancy? Seed dormancy is broken by gibberellin, because gibberellin converts starch into maltose by forming amylase enzyme, and maltose becomes glucose. Using glucose, radicle will become root, plumule will become shoot, and helps in seed dormancy breaking. And gibberellin treatment acts as a substitute for cold season vernalization. Now we have done vernalization. Sir, what is the logic of vernalization? कुछ सीड्स को अगर आप स्प्रिंग में डालो तो वो समर में ग्रो हो जाएंगे, बट फ्लावरिंग नहीं होगा. उसका स्टेम एपेक्स विल पास थ्रू विंटर, और विंटर में स्टेम एपेक्स के ऊपर एक 0 डिग्री सेंटीग्रेड का कोल्ड स्टिम्युलस मिलता है. उससे स्टेम एपेक्स में एक हार्मोन बनता है कॉल्ड एज वर्नेलिन, एंड दैट वर्नेलिन विल गो इनटू द अपाइकल बड एंड हेल्प इन फ्लावरिंग टू टेक प्लेस. तो फ्लावरिंग कब होगा? इन द नेक्स्ट स्प्रिंग. तो वंस अगेन, आपने स्प्रिंग में सीड जमीन में डाला, एंड समर में it grows into a plant with root, with stem, with leaves, but flowers नहीं बनेंगे, flowers नहीं बनता है. Why? Because when this plant will pass through winter, ये है root, ये है stem apex, and ये है stem apex. Winter में there is a cold stimulus of 0 degree Celsius given to the stem apex. उसमें बनता है a hormone called as vernalin, and vernalin will help in formation of a flower. तो फ्लावर कब बनेगा? नेक्स्ट स्प्रिंग समर में यहां पर फ्लावर बनेगा, नेक्स्ट स्प्रिंग समर में यहां पर फ्लावर बनेगा. तो whereas the plant, where was the seed put in? Spring. When did the plant form root and shoot? Summer. But what did the apical bud get a cold stimulus of 0 degree centigrade in? Winter. Winter में होगा. And what hormone is formed? Vernalin. And vernalin will help in flowering. When is the next flowering done? In spring. तो स्प्रिंग से लेके स्प्रिंग तक 2 साल लग जाते हैं, 2 कैलेंडर ईयर लगते हैं. एंड सच प्लांट्स आर कॉल्ड एज बायनियल प्लांट्स. इस प्लांट को क्या कहते हैं? बायनियल प्लांट्स. बायनियल प्लांट्स, दीज आर कॉल्ड बायनियल प्लांट्स. तो व्हाट इज द वे टू मेक अ बायनियल प्लांट बिकम एनुअल? Why don't we put the seed in the spring and इसको समर में ही रूट और स्टेम के साथ फ्लावर बना दो? तो स्प्रिंग का समर में ही फ्लावर बन जाएगा. हाउ कैन वी कन्वर्ट स्प्रिंग इनटू समर? आपको वर्नलिन बनाना है. तो एक तरीका है, सीड को कोल्ड वाटर में डाल दो. सीड को कोल्ड वाटर में डाल दो. व्हाट डज सीड हैव इन इट? रेडिकल एंड प्लूम्यूल. और रेडिकल प्लूम्यूल को कोल्ड वाटर का 0 डिग्री सेंटीग्रेड का स्टिम्युलस मिल जाएगा. एंड रेडिकल प्लूम्यूल, हु फॉर्म्स द शूट एपेक्स ऑफ द फ्यूचर?
Plumule.
शूट एपेक्स इज फॉर्म्ड बाय प्लूम्यूल. तो प्लूम्यूल के अंदर 0 डिग्री सेंटीग्रेड का टेम्परेचर मिल जाएगा. सो प्लूम्यूल विल फॉर्म द हार्मोन कॉल्ड एज वर्नलिन, एंड दैट इज कॉल्ड एज वर्नलाइजेशन. तो प्लूम्यूल में ही वर्नलिन बनेगा, वर्नलाइजेशन हो जाएगा, एंड द स्प्रिंग प्लांट इन समर इटसेल्फ विल फॉर्म द फ्लावर, एंड दैट phenomenon is called vernalization. Another thing you can do is put the seed in the soil in the spring season, and summer में it will form root and it will form shoot. And यहां shoot apex में क्या बनना चाहिए? shoot apex में बनना चाहिए वर्नली. shoot apex में बनना चाहिए वर्नली. And vernalin will help in flowering. और उसके लिए 0 डिग्री सेंटीग्रेड का टेम्परेचर चाहिए. तो what do? We add on the shoot apex gibberellin. Gibberellin helps shoot apex to form vernalin and acts as a superb substitute for vernalization. तो शूट एपेक्स की मदद से ही वर्नलिन बन जाता है एंड हेल्प्स इन वर्नलाइजेशन. तो उससे ही वर्नलाइजेशन हो जाएगा एंड शूट एपेक्स विल फॉर्म अ फ्लावर. शूट एपेक्स विल फॉर्म फ्लावर. एंड gibberellin treatment induces flowering in long day plants under short day condition. तो long day plants को short day condition. Sir, what are long day plants? Summer plants. And summer plants, do we require light greater than critical photoperiod. तो लॉन्ग डे प्लांट्स में याद है लाइट इज रिक्वायर्ड ग्रेटर देन क्रिटिकल photoperiod. लॉन्ग डे प्लांट्स लाइट ग्रेटर देन क्रिटिकल photoperiod. शॉर्ट डे प्लांट्स के अंदर लाइट रिक्वायर्ड इज लेस देन क्रिटिकल photoperiod. So tell me one thing, when do long day plants grow? Which season will long day plants grow?
Summer.
Summer. And when will short day plants grow? जब लाइट कम होती है तो विंटर. समझ में आ रहा है व्यू? तो long day plants grow in summer because light required is more than critical photoperiod, and short day plants grow in winter where light required is less than critical photoperiod. तो समझो आपको एक विंटर में long day plant grow करना है. You want to grow a long day plant in short day conditions in winter. तो winter में light will be less than critical photoperiod क्योंकि light availability winter में कम होती है. तो इनमें फ्लावरिंग कैसे करेंगे? इनमें फ्लावरिंग करेंगे बाय इंड्यूसिंग जिब्बेरेलिन. जिब्बेरेलिन डालते ही लाइट का सब्स्टिट्यूट मिल जाएगा, सब्स्टिट्यूट फॉर लाइट, एंड लॉन्ग डे प्लांट्स और समर प्लांट्स विल ग्रो इन शॉर्ट डे कंडीशन. लॉन्ग डे प्लांट्स ग्रो इन शॉर्ट डे कंडीशंस, एंड हु विल हेल्प इन दैट? Gibberellins. Gibberellins induce growth of long day plants in short day condition. तो golden words, सर हम लोग 3-4 चीजें रिवाइज कर लेते हैं. Who helps in bolting? Gibberellin. Who helps in increase in sugarcane yield? Gibberellin. Which hormone helps in leaf expansion? Gibberellin. Which hormone is more effective than auxin for parthenocarpic fruits? Gibberellin. Which hormone makes Thompson seedless grapes? Gibberellin. Which hormone helps in breaking of seed dormancy? Gibberellin. By the way, seed dormancy टूट जाती है. It can be used in malting process in brewing industry. Malting process in brewing industry. What is brewing industry? Making of alcohol. Brewing industry is making of alcohol. और alcohol में we use gibberellin. क्योंकि gibberellin will break down starch into maltose and glucose. Then fermentation can be done and ethanol can be made. So malting process in brewing industry. समझो आपको विस्की बनानी है तो विस्की के लिए वीट यूज करेंगे, या वोडका बनाना है तो उसमें पोटैटो यूज करेंगे, पर उसमें तो स्टार्च होता है. तो स्टार्च तो अल्कोहल नहीं बना सकता है, तो आप पोटैटो पे स्टार्च पे क्या डालोगे? जिब्बेरेलिन. एंड व्हाट विल जिब्बेरेलिन हेल्प इन फॉर्मेशन ऑफ amylase? What will amylase convert starch into glucose and maltose? And then yeast is put, and yeast will convert glucose into ethyl alcohol. So that is called as brewing. And उसके लिए पहले चाहिए malting, maltose बनाना. तो malting process. So who will help in malting process for brewing industry? Gibberellins. Gibberellins. So seed dormancy is broken by gibberellin, and malting process in brewing industry is done by gibberellin. Who acts as a substitute for 0-degree temperature vernalization? Gibberellin. Who acts as a substitute for light, helps in growth of long day plants under short day conditions? Gibberellins. And gibberellins help in formation of male flowers. समझो आपको एक प्लांट के ऊपर मेल फ्लावर फीमेल फ्लावर दोनों ग्रो हो रहे हैं, एंड द फीमेल फ्लावर इज शॉर्ट, छोटा होता है, Male flower has got beautiful colors, excellent petals. तो commercially important कौन सा plant है? Male. For example, gulmohar. For example, jasmine. इनके अंदर मेल फ्लावर में कलर होता है, मेल फ्लावर में पेटल्स के अंदर नेक्टर होता है, एक्सेट्रा. तो आप क्या करोगे? प्लांट के ऊपर जिबरेलिन स्प्रे करोगे एंड ऑल द फ्लावर्स विल बिकम मेल फ्लावर्स. तो आपको सब मेल फ्लावर मिलेंगे, वो आप पिक आउट कर सकते हो. एंड जिबरेलिन इंड्यूस फॉर्मेशन ऑफ मेल फ्लावर्स in gulmohar, jasmine, etc., where the beautiful male flowers with petals, etc., there. तो horticulture में इसे use करते हैं. In horticulturing it is used. Then we have cytokinins. Cytokinins stimulate cell division, that is cytokinases, and hence they are called cytokinin. Skoog and Miller discovered cytokinins while working on टिश्यू कल्चर एंड टोबैको. तो स्कूग एंड मिलर, सर कुछ साइंटिस्ट के नाम याद रखने हैं. ऑक्सिन किसने डिस्कवर किया था? वेंट एंड डार्विन. ऑक्सिन, वेंट एंड डार्विन. जिबरेलिन किसने डिस्कवर किया था? कुरोसावा, हु वाज अ फार्मर. एंड साइटोकाइनिन किसने डिस्कवर किया है? स्कूग एंड मिलर ने. स्कूग एंड मिलर ने. And during tissue culture and tobacco, they also isolated the compound from autoclaved herring sperm DNA. तो herring क्या होती है? Fish. और fish के sperm के DNA में से मिला a hormone called as kinetin. A hormone called kinetin made from fish sperm DNA. तो ये कौन सा हार्मोन हो गया? एनिमल हार्मोन. एनिमल हार्मोन. और सिंथेटिक साइटोकाइनिन. सिंथेटिक साइटोकाइनिन कौन सा हो गया? काइनेटिन. सिंथेटिक कौन सा हो गया? काइनेटिन. और मिला कहां से? डीएनए में से. सो विच हार्मोन इज मेड फ्रॉम न्यूक्लिक एसिड डीएनए? साइटोकाइनिन. Cytokinin is made from nucleic acid, that is DNA, and they are derivatives of purine nitrogen base called adenine, and they are naturally synthesized by the plant zeatin from corn kernel and coconut milk. तो काइनेटिन किसमें से मिला आपको? हेरिंग स्पर्म डीएनए, which is animal cytokinin. और कोकोनट मिल्क और कॉर्न कर्नल में से क्या मिला? जियाटिन. जियाटिन इज प्लांट साइटोकाइनिन. So which is the animal cytokinin? Animal cytokinin is kinetin. And which is the plant cytokinin? Zeatin. ये याद रखना. And what is the role of cytokinin? They induce cell division, along with auxins, induce cell division even in permanent cell. So a permanent cell can become meristematic. Permanent cell can become meristematic. In other words, differentiated cells can become de-differentiated. Differentiated cell can become de-differentiated with the help of cytokinin. Permanent cells can become meristematic with the help of cytokinin. A balanced combination of cytokinin and auxin is used for organogenesis. Auxins help in root formation and cytokinin in shoot formation during tissue culture. ये हमने अभी अभी किया था. Equal dose of auxin cytokinin form callus. Extra auxins help in root. Extra cytokinin help in shoot. And very importantly, chloroplast formation is initiated by cytokinin in the presence of light. तो एक लीफ के अंदर chloroplast divide. So if you remember, chloroplasts have their own DNA, which is called plastidome. And जब उसके ऊपर light गिरती है, the number of chloroplasts will increase. And that means chloroplast division is done by cytokinin. Chloroplast division is done by cytokinin. In other words, rate of photosynthesis will increase. So rate of photosynthesis is increased by cytokinin. Rate of photosynthesis is increased by cytokinin. And cytokinin delays senescence. What is senescence called? Degradation of organ. And senescence, cytokinin delays onset of senescence by preventing degradation of metabolite or promoting their synthesis. This effect of delaying senescence is called Richmond-Lang effect. So, sir, leaves normally कुछ time के बाद they undergo senescence, that is degradation, and then they undergo falling off, that is called abscission. Which hormone prevents senescence? Cytokinin. Cytokinin helps in mobilization of nutrients. Mobilization of nutrients and delays senescence. तो क्या होगा? समझो ये स्टेम है, स्टेम के अंदर ये लीफ है, लीफ के अंदर उसके मेटाबोलाइट्स कम हो जाते हैं, तो स्टेम के अंदर से एक्स्ट्रा मेटाबोलाइट लीफ में पास होते हैं थ्रू द पीटियोल. एंड जैसे ही मोर मेटाबोलाइट्स आर मोबिलाइज्ड, mobilization of metabolites is done. In other words, nutrients. Metabolites means nutrients. More nutrients are metabolites are put into leaf, the delay of senescence takes place. So delay of senescence takes place, which is called Richmond-Lang effect. And cytokinin reduces the apical dominance and stimulates the growth of lateral bud. ये हमने किया ही था. ये समझो ऑक्सिन है. Auxins will prevent the growth of lateral buds, which is called apical dominance. तो समझो आपको lateral branches grow करने हैं, lateral branches grow करने हैं, then what will you do? You will cut off the apical bud. Apical bud cut off करते हैं, which is done in tea plantations. क्योंकि टी प्लांटेशंस में आपको लैटरल ब्रांचेस चाहिए. टी प्लांट तो बड़ा नहीं होता है, उसके जो ब्रांचेस होते हैं, ब्रांचेस के ऊपर टी लीव्स आते हैं, ब्रांचेस के ऊपर टी लीव्स आते हैं. तो टी प्लांट कोई बड़ा नहीं होता है, उसके ब्रांचेस पे लीव आते हैं, तो उसमें यू कट अवे द अपाइकल बड. इसे प्रूनिंग कहते हैं. व्हाट इज दैट कॉल्ड? प्रूनिंग. Cutting away apical bud is called pruning. तो ऑक्सिन का अपाइकल डोमिनेंस चला जाएगा, ऑक्सिन का अपाइकल डोमिनेंस चला जाएगा, and cytokinin will help in formation of lateral bud. Cytokinin help in formation of growth of lateral bud into branches. तो टी प्लांटेशन में क्या क्या यूज करते हैं? टी प्लांटेशन में ऑक्सिन्स एंड साइटोकाइनिन्स आर यूज्ड. टी प्लांटेशन में what is apical bud preventing lateral growth called as? Auxins preventing lateral growth called as apical dominance. Apical dominance. And who prevents apical dominance and helps in formation of lateral buds? Lateral buds are promoted by cytokinins. Cytokinins oppose apical dominance. Cytokinins oppose apical dominance. And cytokinins help in the growth of interfascicular cambium during secondary growth of plant. Therefore, the diameter or the girth of a tree increases. ये सबसे crucial point है. Which hormone helps in growth upwards? Upward growth कौन कर रहा है? Auxins. And that is called phototropism, towards light. Phototropism. अभी सर lateral growth कौन करेगा? ऐसे lateral growth कौन करेगा? Lateral growth is done by cytokinins. And by the way, what is upward growth called as? Primary growth. Upward growth is called primary growth. What is lateral growth called as? Secondary growth. Secondary growth. So who will do secondary growth or lateral growth? Cytokinin. Who does primary growth? Phototropism, auxins. Who will do lateral growth or secondary growth? Cytokinin. Can you tell me which meristem it will act on? Cytokinin कौन से meristem पे act करेगा?
Secondary or lateral meristem?
Lateral meristem. Which are they? Interfascicular cambium and cork cambium. Interfascicular cambium and cork cambium पे cytokinin act करता है. Interfascicular cambium and cork cambium. What will interfascicular cambium form? Secondary xylem and secondary phloem. Secondary xylem and secondary phloem. तो उससे the vascular bundle size will increase. तो medulla बड़ा हो जाएगा. And what will cork cambium form? Cork cambium will form secondary cortex, secondary cortical cells, secondary cortical cells. So उससे bark of the tree or cortex of the tree will grow. तो interfascicular cambium पे grow करके secondary xylem, secondary phloem से the vascular bundles grow and cork cambium से cell division से secondary cortical cells grow and that will help the girth of the tree to increase. तो ये बहुत बार पूछते हैं which hormone increases diameter of a tree? Cytokinin. Cytokinins grow the diameter of a tree. Which hormone grows apically? Phototropism, auxins. Which hormone causes secondary growth of the tree? Cytokinin. Which hormone causes apical dominance? Auxin. Which hormone opposes apical dominance? Cytokinin. Which hormones help in root formation? Auxin. Which hormone help in shoot formation? Cytokinin. Which hormone helps in chloroplast formation and increase in rate of photosynthesis? Cytokinin. Which hormone causes delaying of senescence? Cytokinin. Which hormone induces cell division along with auxins even in permanent cells? Cytokinin. So which is the main hormone for cell division? Cell division का main hormone कौन है? Cytokinin. By the way, cell elongation का main hormone कौन है? Auxins. Auxins. Cell elongation is done by auxins. Cell elongation is done by auxin. तो याद रखना, cell elongation is done by auxins. Cell division is done by cytokinin. Cell elongation is done by auxin. And who helps in bolting? Bolting कौन कराता है? Internodal elongation. Gibberellins. हाँ, ये याद रखना. And then you have ethylene. Ethylene is a gaseous hormone, colorless hydrocarbon gas. It is hydrocarbon and it is produced by ripening fruits. Methionine से बनता है. Carbon dioxide has an inhibiting effect on ethylene. And carbon dioxide has an inhibiting effect on ethylene. Application of ethylene helps in breaking dormancy of peanuts and buds of many species. Application of ethylene helps breaking the dormancy of seed peanuts and buds of many species, overcomes dormancy of potato tubers. Potato tubers, peanuts का dormancy तोड़ता है. It causes horizontal growth of seedling, swelling of axis and apical hook formation in seedlings. तो ये तीन इफेक्ट है, ये ध्यान से सुनना. So it causes, ये है plumule. Plumule ऊपर ग्रो हो रहा है towards light, ये है radical. And who will help radical to form roots and plumule to form shoots? Auxins. ये auxins करेगा. But plant will start growing, but seedling के अंदर lateral growth भी तो होना चाहिए. लैटरल ग्रोथ भी होना चाहिए, अदरवाइज सिर्फ रूट और शूट ग्रो होगा, सीडलिंग विल फॉल डाउन। तो ये लैटरल ग्रोथ कौन कराता है? If you remember, lateral growth actually should be done by cytokinin, but cytokinin तो acts only on interfascicular cambium and cork cambium, और सीडलिंग में तो इंटरफेसिकुलर कैम्बियम कॉर्क कैम्बियम है ही नहीं। तो lateral growth of seedling or horizontal growth of seedling is done by ethylene. Ethylene helps in horizontal growth of seedling. Ethylene helps in horizontal growth of seedling, swelling of the axis, and apical hook formation in dicots. And ethylene helps in ripening, which is called respiratory climactic. और इथापॉन नाम का केमिकल कमर्शियली यूज करते हैं. So which hormone helps in ripening of fruits? Ripening of fruits is done by ethylene. Ripening of fruits is done by ethylene. And ethylene promotes senescence and abscission of leaves, cotton, cherry, walnut. Senescence, that is degradation, and abscission of leaves is done in cotton, cherry, and walnut. ये याद रखना. It inhibits elongation of stem and roots and dicots. Which hormone promotes growth of stem and root? Auxin. Which hormone prevents the elongation of stem and roots and dicots? Ethylene. And ethylene causes root hair and root hair formation. तो ध्यान से, root formation is inhibited. Root formation is inhibited, but root hair formation is promoted by ethylene. तो रूट के ऊपर रूट हेयर जो आता है वो कौन कराता है? ethylene. Root hair promotion is done by ethylene. And ethylene causes internodal and petiole elongation in deep water rice plants, helping leaves and upper parts of the shoot to remain above the ground. समझो राइस प्लांट्स है, राइस प्लांट ना पानी के नीचे ग्रो होता है, राइस प्लांट वाटर के नीचे ग्रो होता है, और ये है लीफ ऑफ राइस प्लांट. तो लीफ ऑफ राइस प्लांट इज बीनीथ. तो सर, हाउ विल द लीफ डू फोटोसिंथेसिस? उसके लिए कार्बन डाइऑक्साइड चाहिए, और लीफ तो नीचे है. तो ethylene causes the cell division of the nodes, and node will rapidly become internode, and node will become internode and push the leaf outside the water. तो लीफ को पानी के नीचे से लेके पानी के ऊपर लेके जाता है by undergoing rapid cell division and internodal elongation, and push the leaf outside the water so leaf can start doing photosynthesis in rice plant. So internodal and petiole elongation is done. Ethylene causes downward rolling of leaves, which is called epinasty. It promotes female flowering in cucumber. Which hormone did male flowering?
Gibberellin.
Yeah.
Gibberellin. Which hormone causes female flowering?
Ethylene.
Ethylene. Ethylene. And it promotes flowering and fruit setting in pineapple and mango. A pineapple is what kind of fruit? Morphology में किया है.
Composite.
Composite fruit. So entire inflorescence with all the flowers will become one single fruit. याद है कि नहीं, all of you? All flowers with inflorescence become a single fruit, and that is called pineapple. So composite fruit. So who converts entire inflorescence into a single fruit? That entire inflorescence into a single fruit of pineapple is done by ethylene. Ethylene helps in that. And ethylene also helps in wound healing. Ethylene helps in wound healing. समझो बहुत जोर से हवा आई है, and once part of the stem breaks off, एक ब्रांच गिर जाता है, तो जाइलम फ्लोएम एक्सपोज्ड हो जाएगा, तो उसके ऊपर हीलिंग होना चाहिए ना. तो न्यू epidermis xylem should be formed. Who helps in wound healing? Ethylene helps in wound healing. Ethylene helps in wound healing. ये याद रखना. And abscisic acid, it is a growth inhibitor which is called as a stress hormone. It is a growth inhibitor which is called stress hormone. Which is the stress hormone? Abscisic acid. And it prevents germination. So it is called anti-gibberellin. Which hormone breaks dormancy with the help of amylase and helps in germination? Gibberellin. Gibberellin helps in germination. Who prevents germination and induces dormancy? Abscisic acid. So it is called anti-gibberellin. And anti-gibberellin hota hai, abscisic acid is formed in mature leaves, flowers and fruits. बाकी दूसरे हार्मोन young leaf flower fruit में आता है. Young leaf flower fruit, because they are helping in cell division. But abscisic acid grows in mature leaf flower and fruit because it will only help in abscission. वो mature leaf flower fruit को गिरा देता है. So it helps in abscission. And abscisic acid तीन टाइप के होते हैं, inhibitor B, abscission 2 and dormine, of which dormine induces dormancy. Abscission 2 causes abscission, that is falling of the leaf and fruit. And inhibitor B causes inhibition of cell division. So inhibitor B cell division कम कर देता है, abscission 2 leaf, fruit, flower को गिरा देता है, which is called as abscission. And dormine prevents germination, induces dormancy in a fruit. And it's a growth inhibitor. And what is the role of abscisic acid? It causes dormancy in bud seeds tubers during drought. समझो drought है, there is no water available. पानी अगर available नहीं है, what will happen if the seed germinates without water?
Not survive.
The seed will not survive. So abscisic acid under drought condition will not allow gibberellin to produce amylase. Gibberellin high hai, but abscisic acid opposes gibberellin production of amylase, so starch will not become only glucose. और जब तक starch glucose नहीं बनता है, radical का root और plumule का shoot ही नहीं बनेगा. So it will cause anti-gibberellin effect and prevent germination from taking place. तो germination होगा ही नहीं. Abscisic acid retards growth and acts as a growth inhibitor. Sir, plant को growth के लिए क्या चाहिए? Water. Cell elongation करना है, उसमें water चाहिए. अभी if there is a drought condition, तो retardation of growth करना पड़ेगा. So plant will stop growing. That is done by abscisic acid. Who causes growth? Auxin. Who does bolting? Gibberellin. Who does lateral growth? Cytokinin. Who prevents growth?
ABA.
Abscisic acid. Abscisic acid will prevent growth. And abscisic acid is found in the leaves and during drought causes closure of stomata. Closure of stomata. Why should stomata be closed?
Excessive photosynthesis.
To prevent transpiration. एक तो drought condition है, temperatures are high. अगर stomata खुले रहेंगे, तो plant will lose water and that will cause transpiration. Can you tell me which hormone opens stomata? Closure of stomata done by abscisic acid to reduce transpiration. Who will open stomata?
Cytokinin.
Why? Very good, Pradnya.
Sir, I guess because it causes the chloroplast formation, so the it will increase, the number of chloroplast will increase, so that would help in the iron to come inside and then photosynthesis.
Correct. If you remember, cytokinin increases number of chloroplasts, means rate of photosynthesis बढ़ जाएगा. अभी rate of photosynthesis बढ़ाने के लिए we need more carbon dioxide for dark reaction. So stomata will open. So cytokinin opens stomata. Cytokinin opens stomata to increase rate of photosynthesis and abscisic acid closes stomata to prevent transpiration. ये याद रखना. And abscisic acid promotes senescence in the leaves, causing loss of chlorophyll and decreases the rate of photosynthesis. Who has it opposed? Who prevented senescence?
Gibberellin.
cytokinin.
Richmond-Lang effect. याद है कि नहीं? Who caused production of chlorophyll?
cytokinin.
And who increased rate of photosynthesis?
cytokinin.
Who opposes cytokinin?
Abscisic.
abscisic
Acid. It promotes senescence in leaves, causing loss of chlorophyll, decreases the rate of photosynthesis. क्योंकि drought condition है, drought condition में leaves जितना जल्दी हो जाए गिर जाना चाहिए. Leaves should fall off. नहीं तो rate of transpiration is high. So it will break chlorophyll, it will cause falling of the leaf, promote senescence, exactly ulta of cytokinin. It inhibits RNA and protein synthesis, and under stress condition like drought, flooding, mineral deficiency, it overcomes stress by promoting abscission. So abscisic acid causes dormancy. By causing dormancy, it opposes gibberellin. Gibberellin breaks dormancy and helps in germination. So it will act as a anti-gibberellin. It retards growth. Who helps in growth? Growth is done by auxins. Root and shoot growth formation is done by auxin. It retards growth. So it is antioxidant. It helps in closing of stomata. Opening of stomata is done by cytokinin, and it helps in closing of stomata. So it is anti-cytokinin. It promotes senescence, loss of chlorophyll, decreases rate of photosynthesis. Everything is done by cytokinin, and it will oppose cytokinin. It inhibits synthesis of RNA and protein. Normally, RNA and protein synthesis is done by auxins. So it will prevent auxin or oppose auxin by inhibiting RNA and protein synthesis. It promotes abscission. Which hormone had opposed abscission? By acting on abscission zone. Auxin. याद है, auxin prevented young leaves from falling. Auxin prevented young leaf from falling. Who will oppose auxins and promote abscission? Abscisic acid. Have you understood this, all of you? Do you all have a test right now?
Yes, sir.
कितने बजे बेटा?
It was on 9 o'clock, like 9 to 10.
अच्छा, okay. चलो, तो ये हमने abscisic acid कर दिया. You understood everything, all of you? चलो, all the best, dear. Take care. See you on next Tuesday or next Thursday.
Sir, एक छोटा doubt था. In cytokinin, we can say it promotes hedge formation, to be more precise, for tea and coffee plantations.
Yes, beta. Promotes hedge formation. Correct.
Okay,
Correct. Chalo. Okay. Give your test, guys. Take care. Chalo. Bye-bye. All the best.
Bye, sir.
Bye.
Bye, sir. Thank you, sir.
Bye, beta.